I'd like to say thank you for this great invitation. It's my first time in Indonesia. I'd like to thank the Prof and Steve for arranging this. This is all new to me, as some of the stuff I will be talking about to you I'm sure will be new as well. So I'm just going to go quickly through several slides. What I want to do is generally give you an idea about what an editor does at a nature journal. And I presume everybody here will know what nature is. But, you know, nature is seen as this kind of monolith. It's, you know, to everybody outside of nature, it's one thing, nature. I must get published in Nature. But, you know, there are many different flavors of nature journals, and I'm going to talk to you a little bit about that. And then I'm also going to talk to you a little bit about my journal, which is Nature Biotechnology. And then I'm going to give you a brief run through some of the exciting things that are happening in the biotechnology space. And then finally, I'm going to give you a few practical tips if you ever get into the position that you want to send a paper, whether it's my nature journal or one of the other nature journals, because there are certain principles that apply to all of them, and I'm going to go through that. So, firstly, I'm going to what an editor does. Then I'm going to talk about nature biotechnology itself. Then I'm going to go through the recent trends, and then finally I'm going to tell you how you get your nature paper. So I wonder if this is all recognizable to you all. This is so this is the Gladiators Forum, yeah? And you know, these kind of combats used to take place in antiquity, and at the end of the combat, when there was one victor, essentially he would look up and there would be a thumbs up or a thumbs down. So does anybody know who would give the thumbs up and the thumbs down? This is really interesting because my thought was that it was actually the emperor, yeah, who said, you know, kill him, rather than, or let him live. But it was this person here, and that person was called the editor, yeah. Isn't that interesting? It has been pointed out to me that this kind of thumbs up and thumbs down for papers has many parallels in the publishing universe. So when I joined Nature Biotechnology back in 1996, a very long time ago. So I'm quite ancient, and at that time I had an idea that, you know, I was leaving academia, I was going to go to Nature, and, you know, this would be a great job. I'd read a few papers in the morning, have a cup of coffee, you know, maybe go out for lunch, have a late lunch, type a few letters, read another paper, maybe, reject a few more, and then go home. So there I am. But in reality, it's not like that at all. So one of the things that you need to remember in today's world for editors is that the most precious thing that an editor has is time. We are under constant pressure to do everything that we do under, you know, faster and faster. And it's like science. This is like everything in society, yes? We're trying to do things faster and faster. And faculty, PIs who are, you know, writing papers, they're scared of being scooped. They want their paper to get through the process faster so that their competitor, you know, doesn't get published before them. And so this is getting, you know, there are more and more journals, there are more and more papers, there are more and more scientists. And so this is a continuous, you know, pressure that's on everybody in the process. So everything in the process has to be about how do I use my time most productively. And so what are the things that I do as an editor? Yeah? So this is the key, this is the core of the job, is that the manuscripts that are sent to us by researchers. We read the papers, and then we decide which papers we're going to send out for review. And the editors are meeting every day, and we're thinking about papers. When we read the papers, I'm going to talk about this a little later, but the way it works is the papers are shared out to editors who specialize in certain areas. They read the papers, and then they come to a meeting. Each day there's a meeting where all the editors and yours truly is present, and the papers are all discussed, and each of the editors presents their paper, and they'll make an argument for why it should be sent out for review or why it shouldn't be sent out for review. And sometimes after the discussion, the decision, if the editor might have come and thought, well, I don't think this is good enough, but the discussion between the editors changes people's minds, and then we think, oh, okay, we are going to send this out. Alternatively, and the opposite happens. Somebody comes and they're like, oh, I'm really excited about this, and then one of the other editors, brings up previous work that makes it less exciting, and so on and so on. So it's a consultative process that goes on with each paper, and we're constantly, as we're publishing papers, we're constantly thinking about, so if we publish the next paper in this area, where do we set the bar? Because we don't want to publish a paper that doesn't advance from where we are. So we're constantly thinking about, what are the criteria that we should be applying to the field. The second thing that's really important and is a big part of my life and what I try to do for my editors, which again comes back to time, is to send my editors out to different universities to do what we call our lab visits, where the editors will come to the university, they will talk to the faculty, and the faculty will discuss the things that they're working on. And very often there will be a discussion between the faculty member and the editor, and the editor might give some feedback. Perhaps you might not want to do this type of experiment. This might be a better way to go because I saw this paper that was published, and it's this example of it's a dialogue between the two sides, and this is very important for us as well, because there's nothing worse for faculty, I think, to send a paper to a journal that's like a black hole. Yeah, you send the paper there and then it disappears. Yeah, and who do I contact? So these types of visits where the faculty gets to know the editors, and I encourage all the faculty to try and do this with as many editors as you can, is to attract them to come here and to talk to you. It's really important. So another part of our job is just editing the papers. So even English language speakers don't write very good English sometimes. So we spend some time. Whoops, losing my head there. We spend some time kind of editing the research papers, and we also are always on the lookout for new areas of research, and maybe my journal isn't receiving papers in a certain area, and I want to go out to the community and say, listen, we're interested, send your papers to us. So if that's the case, then we do what are called reviews, or news and views, where we ask an expert in the field who is recognized in that area to write a kind of summary of what's going on and why it's exciting. And then that signals to the community that even though the journal might not have published in that area, we are interested in this type of research. And then finally, this is very interesting. As time has progressed, we spend less time on outreach to the media. And one of the reasons for that is we found that the media don't really listen to, what we think are the most important papers and what are they just, there's no, and it's got worse and worse. the types of science that's highlighted by the media and the types of good science that's just ignored. And I think part of the problem is that the media, a lot of the science specialists in the media are disappearing. Most of the best science journalism now is on the internet in terms of blogs and stuff like this. It's very difficult to find really good science coverage on the internet. So we do write press releases occasionally, especially if we think that the press are going to misunderstand a paper, but we don't spend much time on this anymore. So again, just to emphasize the point that it's nature is a village, that there's a lot of journals now. So when I came to Nature Biotechnology in '96, there was the mothership here, Nature, which has been around for quite a long time. And then in 1992, Nature Genetics was the first journal that was launched, and then all these other journals have been launched since. In 2011, this journal was launched, and actually this is quite an important breakout journal because it's an open access journal. All these other nature journals are not open access, and we can talk a little bit about why they're not open access at the end. And then there are these, again, these nature reviews journals that I'm sure you're familiar with. So just to talk a little bit about my journal, Nature Biotech, it's a little different from most of the other nature journals, which are more basic. So Nature Biotechnology is an applied journal, and we're also a journal. We're kind of a bit of a weird journal, to be honest, yeah, because no other scientific journal thinks about business. I can't really think of one, yeah, and yet, if you look across academia, across the world, all the deans, you know, they're saying to their faculty, come on, we need to get involved with industry and do this. And yet, you know, how does faculty learn about this if none of the places where faculty, you know, read talk about it? Yeah. So I think this is really important that we always try and do as good a job as we can to describe some of the difficulties in this. And, you know, from the scientific standpoint, we're a bit weird as well, because nobody's really a biotechnologist. You have geneticists, you have bioengineers, you have engineers, you have chemists, you have material scientists, but there are very few people who would call themselves biotechnologists. So we are a journal that doesn't really have, you know, one specific field of researchers who's reading us, yeah? All the different fields have an interest in us, but we aren't really their journal. And Nature Methods has a kind of similar problem. And, you know, just to emphasize that this magazine part of Nature Biotechnology is quite different from a lot of the other nature journals. Okay, so I made that point. So these are some of the areas that we cover in what we call biotechnology. There are 20 areas in all. There are 5 editors who work on the journal. So each of those 5 editors specializes in 4 areas. And when the paper comes in, it's sent to that editor, and that editor reads it. And the editor probably has about an hour to read the whole paper, search through the literature, you know, maybe look up a few faculty on the internet, think about the paper, think about how to write it up, what are the pros, what are the cons, before they come to the meeting. So that's all that they've got, and I want you to think about that, because when I come to the end, it's important to how you present your research, to have the greatest traction with the editor, so the editor understands the importance of what you're sending. So again, this kind of speaks to the fact that we have competitors, but they're not really, they're in different bits of biotechnology. So we have materials, a lot, we regard a lot of these nature journals as our competitors, and this is another important point to make, that with some of the cell journals, the papers go to Cell and then they go to Neuron or Developmental Cell or Cancer Cell. All of the nature journals, we're competing with one another, and that's because competition makes you lean and mean, yeah. And so we're looking for papers, and we also even try and nick papers from Nature, yeah. So it's really is a competition, and that's what kind of makes it a lot of fun. So here this is the latest Nature journal, Nature Plants, and obviously plant biotechnology is another area of great interest to the journal. So when I took over the journal in 2000, we had an impact factor of 11.5, and for what it's worth, it's grown since that time. One important thing to say about impact factors, they're a tyranny. Yeah, it's unfortunate that they are kind of used in the way that they're used, and I think there are other metrics of productivity for scientists that are much better than impact factors. But these numbers strike the fear of God through any editor. When it comes around May or June of the year, these numbers come out in the summer. If these numbers are bad, then that's very bad news for us, yeah, when we're going around for looking for papers. Okay, so that's just a bit of brief background. I'll now talk a little bit about the submissions that we receive. So we think about the area in these kind of 4 broad categories. I'm not going to talk much about these 3, agriculture, energy and environmental, but you can see how dominant the number of submissions that we receive from the biomedical arena. It's like 80% of all the submissions that we receive, and in effect this follows the way a lot of funding is. I mean, if you look in the United States, for instance, the NIH gets a big whopping, high 20s, low $30 billion, and the Department of Agriculture is kind of down in like 7 or 8 billion or something. So, there's no magic to these numbers. I think it just reflects the fact of where the funding's going. This is how many manuscripts we receive per month. So we receive around 150 manuscripts. This is a scary slide as well, yeah? I can understand that this is quite daunting, yeah? The fact that only 20% of those papers are sent out for review. So I'm not going to argue with you. It's really hard. It really is hard to get into these journals. And again, about 20% of them are published in the end. We try, so each editor's handling around 25 new manuscripts per month, and in terms of decisions, we try and give a decision in a week from when we've received a manuscript. Okay, so this is a slide just to kind of talk about which institutions are producing the papers, and if you look at that, then it really is dominated by US institutions. You have a few UK institutions. The Chinese Academy of Sciences also does well. But the takeaway message is this still is dominated by the original, players in terms of the number of papers that are coming in. Now, in this slide, what we did was because we're often asked, well, is there a bias at Nature in terms of publishing papers from the UK and the US compared with other places in the world? And even though the number of submissions, 80% of our submissions are from the US, you can actually see that the US doesn't do as well as some places in the world like Scandinavia or the Netherlands or Switzerland. Spain does well. And this is kind of interesting. Latin America does quite well as well. So if you look at the median ratio for, this is all of those 18 Nature journals, and you kind of work out, you know, what the submitted to published ratio is. It's around here. So this is just to show that just being at a US or UK institution doesn't mean that you're going to do particularly well. So I looked through my database, and you'll be interested to see that I found Indonesia there. There we go. So I think the take-home message is we need more submissions from Indonesia, okay? If there's one takeaway message from my trip here, we want more submissions from Indonesia. Okay, so into the last two-thirds of the talk. Actually, last half of the talk, sorry. So I'm going to talk a little bit about some of the areas where we're seeing really interesting research happening around the world. So you've probably hearing a lot about genome engineering and synthetic biology and some of these new molecular biology approaches. And this really is starting to open up several areas of interesting research around the world. This made a big splash 2 or 3 years ago. You might remember J. Craig Venter, who was one of the people who worked on sequencing the human genome. He then moved on to try and create an organism from first principles. And this caused a big splash in the media when he essentially was able to synthesize a whole genome and then take that genome and put it into a different species of organism and turn that organism into a new life form. Yeah, and so the media got all crazy about this. But it's a really interesting approach, and it suggests that there might be ways of harnessing this in the future. But there's very few labs that are working on this, and it's actually pretty hard to do. But different people are taking different approaches to engineering genomes. One can minimize genomes by taking out non-essential elements. One can fuse genomes of different organisms. One can redefine codons. So you can change the codon content of genomes. One can create libraries of synthetic DNA that one then transforms into different cells and then screens those cells for different activities. And, you know, we're starting to see, you know, ways of creating in vitro expression systems that are outside of the cell. But these are all kind of very experimental new approaches, and I think in the next few years they're going to have important applications in areas like metabolic engineering and essentially other approaches to creating. I think they're really going to have an impact in the world of microbiology, so creating new types of prokaryotic organisms. And one of the reasons that these technologies are being opened up is because DNA synthesis has become so much cheaper than it used to be. The fidelity is much, much better. We published these 2 papers on array-based oligosynthesis. I think the days of cloning, which we all did in the 80s, 90s, and 2000s, is going to, you know, ultimately I think we're going to get to the stage when everybody just orders your gene from a synthesizing company. I think that day is very close. This is another interesting area. This was a paper that was published a couple of, actually 3 years ago now. The thought that one doesn't have to just have the 4 bases that we've been talked about as the formation foundations for life. One can start to engineer new types of nucleic acids, which means that one can create organisms that are what we call orthogonal to natural organisms. And what that means is, like, if you can create an organism that is orthogonal, that won't interact with a natural organism, you can start to think about, well, maybe I can put these organisms into the environment and there will be no possibility of any interaction with nature at all, because there's no way for these organisms to survive by breeding with natural organisms. So there's a lot of excitement about this. And more recently, this paper was published where it's been possible to create an organism that's a living organism that has a different genetic alphabet. Another area that you've probably heard about and is very exciting is this area of gene editing. And gene editing has been around with us for a few decades now. It started out with zinc finger nucleases. More recently, talons came to the fore. But since 2012, really, the world has been taken over by this technology called CRISPRs, which is a prokaryotic system. And one of the key things about this system compared with the other types of gene editing system that I showed you earlier, these are proteins. And when you're changing the code here to bind your sequence of interest, you really have to do protein engineering, and it's quite tricky. But with CRISPRs, the whole process is mediated by the guide RNA. And what that means is, it becomes, to target the gene of interest, it becomes as simple just to, design a nucleotide sequence. And that's very much easier than engineering a whole set of proteins, and makes it work much faster. And we've just seen this explosion of papers. I mean, Nature Biotechnology is almost Nature CRISPRs these days. There's so many papers that we're publishing in this area, and the area is moving very fast. And one of the interesting things about that, I think, is that you see this with technologies that really work. They spread like wildfire through the community, and that's because when people use them in their hands, it immediately works. Yeah. So I think this is a really promising development, and there are all kinds of areas where these things are going to be important. People talk about using gene editing therapies in the context of gene therapy. So in medicine, you can think about certain rare diseases, monogenetic diseases, where you could make a change to remove a deleterious allele with a CRISPR. But there are also, really exciting applications of this stuff in research. So, for instance, if you can start to use CRISPRs in the process of making genetically engineered mice, you can reduce a process that used to take 2 years down to 3 weeks. So this is enormously empowering. Yeah? And if you think about some of the things that have been happening in the past few years, in the whole of the biotechnology arena, which is, we've been sequencing genomes, learning gene content. We now have this tremendous library of genetic content that we need to know the function of. And really, CRISPRs, this allows you to start putting those genes into animals and asking the question, and you can do it really, really fast. This is why the community is really excited. Yeah. Another area that we cover is sequencing, which has been very important for both in clinical research and in basic research. And there are all kinds of different platforms. I don't really have time to go through this. But needless to say, I made this slide about 2 or 3 years ago, and some of these technologies no longer exist. They're kind of obsolete. It's so fast moving. This is no longer available. Helicos is no longer available. There's a very, very different Illumina is one of the main instrument providers, of course, that is dominant in the market. But some of these long sequences, so these are all short sequencing platforms, short read sequencing platforms, and this is a long read platform. And there are ways in which you can combine the platforms together, which allow you to ask interesting questions and validate either platform. And then you've probably heard about nanopore sequencing. So this is another area that has been an area of promise for, I would say again, this kind of goes back to what Steve was saying about it taking 10 or 20 years for something to come to fruition. This technology, there's a company in England called Oxford Nanopore that is now producing devices that are like a stick for your computer. They're that big. They're not like a big benchtop. They're literally this long, and you plug into your computer like your memory stick. And you know, again, it's a leap forward. It's a massive leap forward. It will enable us to do very interesting experiments because it's a long read technology. You can read thousands of base pairs with this technology. And if you combine that with some of the short read technologies, it's going to be an enormously powerful technology. So I'm going to run through some of these quite quickly. All I want to say is sequencing is enormously powerful because you can turn what I would call analog assays, studying chromatin interactions or methylation, protein DNA interactions. You can use a sequencer to what we call digitize the process. And what that means is you can do these experiments much, much faster and with much higher throughput. Again, you know, these are affecting areas, many different areas, whether it's plants, whether it's microbes, whether it's animals. And I haven't really even talked about clinical genomics. And essentially, you know, microbial genomics is becoming a very, very important clinical tool in tracking down outbreaks of epidemics. So it was unthinkable before that one could track down, you know, which pathogen is causing, you know, this disease outbreak of some food pathogen in a matter of, you know, weeks, sometimes just 1 week. A new organism emerges. Nobody's ever seen it before, but we can now detect it using some of these technologies. And I just put a few examples of indications where you know, that's started to become possible. This is another area that's going to completely explode in the next two or 3 years. You've probably heard in the pharmaceutical industry, in the area of cancer, we've had traditional chemotherapeutics, then we had a second wave of drugs like Gleevec and Avastin, the kinase inhibitors, and now there's a new set of drugs that work with the immune system to activate our immune systems to counter cancer. And some of the results are staggering. You know, remissions, people who've been cured of cancer and it's lasted for not months like these other treatments, but years. People, these are kind of melanomas that before it was a death sentence, and you know, now the patients are completely in the clear. But one of the problems is that many of the patients don't respond, and nobody really knows why. And what one can do with sequencing is start to ask, so why do cells in one patient respond, and why do the cells in the other patient not respond? So this is just going to grow this area. I'm not going to linger over all of these areas because we're going to be here for a long time. So I'm going to quickly move to talk a little bit about how to get published, because that's really what you're waiting to hear. Okay, so what do we look for in submissions? So one of the key things that we look for is this idea of conceptual novelty. So this kind of comes back to what Steve was saying in terms of curiosity-driven research. One needs to be asking big questions and getting big answers. One needs to be moving a field forward, and it's not sufficient to repeat what somebody else has done with a little wrinkle, because nature journals want a lot of people to be interested in the papers that they publish, and so that means it has to, a new idea is very, very compelling. We're looking for advances with implications that cross disciplines. So maybe you're working on a problem and it's really important to the people in your field, but if there's nobody outside of your field who's interested, then it's less likely that we're going to be interested, because your field is only a very small part of all of our readers. Then if your research maybe changes a paradigm, a way of thinking, conventional thinking, that a field has kind of thought this is really important, and this is the way it is, and your paper turns that on its head. That's another, it's kind of related to the conceptual novelty. And sometimes you may connect areas of research that nobody's made connections between before. And again, the novelty aspect of showing that a technology can now ask a question in a new system that nobody had shown that system was useful for. So one time I gave this talk, and one of the questions at the end, an academic kind of stood up and said, "Oh, nature journals, it's just trendy, isn't it?" But it's actually correct. I mean, he was quite surprised when I agreed with him. Nature journals really are about a particular type of science. So the important thing to remember is that we reflect you. That's, if you're working in an area that currently isn't really trendy, it is more difficult to get in a nature journal. And I want to emphasize, we reject many papers that are of comparable scientific quality to what we publish. Yeah? So what I want to say is there's a lot of good science that we're receiving that is just as good as the science that we're publishing, but it just doesn't get published because the communities that we're serving aren't telling us, wow, that's what we want. And of course, you've probably, this is a goal for us, and what one might argue that we're not doing as well with this as we have in the past, or we're not doing as well with this as we could have. And there are all kinds of reasons for that we could maybe talk about with at the end if there's questions. So if you're going to prepare your paper for a nature journal, what are some of the things you should think about? And these are called pre-submission inquiries, which is essentially a little abstract, about 100, 200 words, describing your work, what it is that you did, what did you find, and why is it important. And it's literally that. And if there are key citations of work that happened before that your work builds upon, cite those papers. And so the reason for doing this is that we try and give an answer to you much quicker than if you send us a paper. So if you want to get a quick answer, it's a good way of kind of asking a question of the editorial team. So another question we're asked was, so when you send your paper to the journal, another thing that you would do is to recommend people that you would like to referee the paper. Yeah? So very often we receive a list of people who are recommended referees and a list of people who are excluded referees. And so in terms of the recommended referees, we do sometimes use them, but very often it's because they're people that we were thinking of anyway. Yeah. So the fact that the author has recommended that person as well, that makes us think, oh, we were thinking in the right way. In terms of excluded reviewers, so perhaps a competitor, we do all that we can to honor this, because clearly it's important. Although, occasionally, and this is a particular problem in small fields, is that we get like this list that's about this long of people who are excluded, and there's nobody left to find as a referee. And so in those cases, if the list is unreasonable, then we're not going to, we're going to approach some people. Yeah. So I would emphasize it is more important to list people you prefer to exclude, and let us know if there's competition, if there's a paper out there that would potentially scoop you. Okay, then another thing, so the cover letter, this goes with your manuscript that you've sent. The first, and this is important, you remember at the start I was telling you the one thing that we're desperate is for time. So the editor really needs to, is looking at, he or she has many different things to do. And so the important thing is, if you can catch their eye by writing a good cover letter, why not? Yeah? Why not do it? So grab the reader's attention and give them a reason to be interested. Summarize the events and place it in the context of, so what went before and why is this important? Why should the editor be interested in this? And if there's something unexpected or surprising about the results that you had, emphasize that. It's very important. It's a bit like this kind of bottom line. I always think it helps with the paper to really know what you think is the main take-home message of the, because all the best papers, and again I think you said this, Steve, there's a very simple, I think you said it in the way, we're not all geniuses, you said geniuses kind of simplify things down, but it's very helpful if you're clear about where the advance is and why it's an advance, and don't get trapped in the details, because we're not special. The editors aren't specialists. We're generalists. We're like our readership, who are also generalists. So we think in those terms. We think about why is this important for a broad audience, and then try and justify why are you sending the paper to Nature Biotechnology? Very often we receive cover letters to Nature Medicine and Nature Biotechnology or to Science, and it might seem like a trivial thing, but it just shows that you've just sent it out and you haven't really thought about where you're sending the paper. It helps. This sounds trivial, but if there's somebody else who can read the letter and check for spelling or grammatical errors, then why not? And sometimes when editors go out, as I mentioned at the beginning, we go out into the community and we solicit papers, We just don't wait for papers to come in. Sometimes an editor goes out. Sometimes, I will not deny that this happens. An editor goes into the community, solicits a paper, the paper comes in, another editor reads the paper gets rejected. That's not good. That's not good. So it's really important if somebody within the editorial team has spoken to you about the paper, to mention in the cover letter, I spoke to Dr. Marshall about, my work, and he invited me to write in. That will immediately put your paper to the top of the pile, yeah? And, this one is clear. I'm not saying that it happens often, but it does happen. So, let's say your paper's been sent out for review, and it's come back, the editors have got the 3 or 4 referee responses, and then, they've decided to reject your paper. So you come into your office in the morning, and you have this dreadful, after all this work that you've done, you got it sent out, you were part of that 20% of papers, and now it's been rejected. So the your first your first reaction may be to write back to the editor and tell me that I'm an idiot. Yeah? That's fine. I mean, I do receive lots of emails telling me I'm an idiot. But the fact is, it really doesn't. I'm not affected by it because I receive those letters all the time. Yeah. What I respond to, and it's understandable. We all understand that it's really frustrating, and editors understand. We're people like you are. We understand that a whole career has been. You might have spent years working on something, and it's important that we get the decision right, and editors do make mistakes. So what editors respond to is rational arguments. So what we want to know is, we made a decision on the basis of the evidence that we knew from our reading of the paper, and also the questions that we asked the referees to look at in the paper, and we synthesized that all together, and then we made our decision. But sometimes, referees have misunderstood a paper. They've misunderstood a particular set of results. Sometimes they've requested experiments that are completely unreasonable, and an editor, might not be familiar with the field and will, say, okay, well, that sounds like a reasonable. Spend another $1 million doing 2 years' work with those animal experiments. So if something is unreasonable, write back to the editor. Tell us why it's wrong. Yeah, it's really important. We want to know. We want to know if we've made wrong decisions. So always be polite and respond with thoughtful, rational arguments. Point out to us, yeah, if a referee has kind of missed the point, and also, I think it's really demeaning if a reviewer has spent their free time reading your manuscript and critiquing it, if you write back and just criticize the referee, saying they're an idiot, they don't know what they're doing, clearly they're not in the field, blah 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 blah, yeah. These types of responses are not constructive, and I should say as well, I'm here in Asia, and these types of responses aren't the types of responses we usually get from Asian authors. We get these responses from American authors. Oops. So, try and think about, keep it simple and to the point when you write back to us. Respond to all the points that have been made in the letter, and see if there's things that you can compromise. If you ring the editor up and say, I would really like to talk to you about this. I think I can do this experiment, but this experiment is just really not, something that I would like to do. And here's the reason why. Then we will listen. Yeah, because the thing that I want to emphasize is, when we send that 20% of papers out, we are dedicated. We don't want to reject that 20% of papers, because we've made a decision. We want to publish those papers. Yeah? We want them to be the best papers. So it's not only a drawback for you that the paper is. It's a drawback for us, and because the editor has spent time, you know, and the referees have spent time on your paper. I just want to talk to you about a few things that we're doing at the moment in thinking about how we can do our job better for authors and also for reviewers, because the one thing that's important to the editors at Nature is quality. I think there are more and more journals that are, you know, out there in the publishing universe, and what Nature wants to stand for and wants its editorial process to be the best process that there can possibly be. So I speak not only for my team at Nature Biotechnology, but all of the Nature editors are interested in how we can do the job that we do better and stop making mistakes when we're reviewing papers. So some of the things that we're doing to kind of improve the papers that we publish. So here, one thing that I think is really important, talking about young researchers and students and profs and, you know, papers. So one problem I think is that the Nature paper very often makes or breaks somebody's career, yeah? And so it's really important if the person who did the work is properly acknowledged. And very often it really depends on what the culture is of a particular institution, but sometimes that doesn't happen. Maybe a more senior person gets the credit rather than a more junior person. So we started this several years ago, trying to make these contribution statements in papers that indicate clearly who did what in the paper. And I think that's really important. Another thing in terms of making sure that we know who it is that did the work. Right. So there are many Kims in Korea. There are many Marshalls in the UK. And one of the things that one wants to do is to differentiate everybody. So you may not have heard of these, but these are kind of ORCID identifiers, and now all Nature papers have these ORCID identifiers on the author, so we know, you know, you can track that author in the literature and what they did. So here's a big change that, and this is starting to happen across all Nature journals. And I think it's a really important change. So what used to happen is the referee reports would come in, the editors would read all the reports, and then make a decision as to whether to reject the paper or to ask an author to revise the paper, or in some cases to accept the paper. But essentially that was the way the decision was made, and the letter was then sent to the author. Now, what we think is wrong with this is, like, that works for papers where the decision is very clear. But the fact is, in most cases, the decision is not clear, and it's actually really helpful if you send the referee reports back to the author, and then the author can read through them and respond back to the editors and say, well, the referee makes a good point here, but this point here, this is just crazy. I'm not going to spend my whole life doing these experiments, and it's nothing to do with the main claim of the paper. And so, you know, what that means is that we now take that information into account when we make a decision. Again, in terms, I talked a little bit about reproducibility and replication in papers. So what we're trying to do now, there's this new entity called scientific data that allows large data sets associated with papers to be peer reviewed in themselves. So you can actually generate a data set, have that data set peer reviewed, and get a citation in scientific data that you can use like a paper citation. And this allows very high quality data sets to start to get into the literature and to be enabled to be shared. So we're not kind of stuck just with papers, because in this world of high throughput data, what we want to do is open up data to the community and allow these things to start to happen. Very recently, this just happened last month. Actually, where are we? We're March. It happened today for Nature Biotechnology. The journal now is offering double-blind peer review. So those who are wondering what that is, it's essentially the way most traditional peer review works is that it's single-blinded. So the author is not anonymized. The referees know who the author is, but the referees are blinded to the authors. Now, there are a few problems with that, which is, some younger researchers might feel that they would, the fact that somebody's a big shot means that they get better treatment. So isn't it better to have a system where you just get rid of who it is who did the research and just let the research stand for itself, yeah? And so we're offering this service now. So if you're an author and you want to have your paper sent through the double-blind review process, we are now doing that. Last 2 points, referees, and I want to make this point as well, because this is another crisis that's happening in research that's due to the explosion in papers. It used to work really well, like a few decades ago, but now, because there's so many papers, it's like, and I'm sure the faculty here probably sees this yourselves, yeah? Is you are bombarded with requests to review, yeah? And it's becoming really, really difficult for faculty to have the time to review papers, because these requests are coming all the time. So what we want to do is we want to make the process as least painful for the referees as we can. So we want to make sure that when we get referee reports in some cases we might circulate the reports among all of the referees, and so that helps, because sometimes a referee can see another referees and they can contribute to the decision, and it enhances the process. And then, of course, we also acknowledge referees who give us their time for free, more than, who've reviewed for a Nature journal more than 3 times a year. Now, I'm aware I went over time quite a bit, but if we do have times for questions, I'm very happy to oblige. Thank you very much.